in this exciting role. Now I'll hand it over to Sue Baldwin, who's I guess the leader, the director of this research theme within BRIMM. I neglected to mention that BRIMM has a number of research themes, of which this is one, but Sue will play a very important leadership and coordination role in this theme. So fitting that we should finish with Sue.
Okay, great. Thanks very much, John. First of all, can everyone hear me?
Yes, indeed.
Great.
You can see.
And can everyone see my slides?
Yep.
Well, if you can't see my slides,
we
okay, great. Thank you. Okay, so thanks very much for joining our BRIMM webinar today. I'm going to talk about how the microbiome can play a role in mine remediation, with a specific focus on how we can harness and leverage the native microorganisms that are already present on the mine site. So my name is Susan Baldwin. I'm a professor in the Chemical and Biological Engineering Department at the University of British Columbia. My interests are in bioremediation, biomonitoring, use of active and passive treatment for mine impact water, applied metagenomics and microbiology, and the circular economy, the valorization of residues for reuse, and recovery of values from wastes. So these are mined landscapes. These cover very, very large areas that are many square kilometers in size. So during mining, the tops of mountains are removed, large pits are excavated, and enormous storage dams are built. These landscapes are mostly devoid of any life as far as we can see by the naked eye. So not only do we need to restore these landscapes to their natural forms, full of life again, we also must protect the surrounding ecosystems from any deleterious impacts from water runoff or any other emissions from the mine. So unforeseen events can cause catastrophic disturbances on the surrounding, untouched environments such as lakes and rivers that are adjacent to mines. Fortunately, these do not happen very often. So this is an aerial view and a land view of the physical impacts that you can see from the breaching of the Mount Polley tailings dam in 2014. And Stephen mentioned this in his talk. So even though these mined environments and the effluents that flow from them look like they don't contain any living organisms, there are always microbes present. And so Rachel conveyed that really nicely in her talk, showing the microorganisms, or the cyanobacteria, I think it was, that were utilizing iron in the oceans. So microorganisms are everywhere, and they play a very important role in biogeochemical cycling, as we've been hearing about today. They can transform minerals into different forms. They can dissolve solids. They can cause things to precipitate out. They can make acid, as Vikram explained in his talk. They can change the local environment in many different ways, and these can be good ways or bad ways. So one technique that we can use to find out what microorganisms are living in these mine environments and how similar or different they are to the pristine surrounding environments is the application of eDNA. So this stands for environmental DNA, and this is very similar to what Rachel described in her talk, is basically you walk around the mine site to the locations that you're curious about, and you pick up some soil. And similar to Rachel's pictures, you just scoop some of the soil up into a bag, a sterile bag, and then you take that back to the lab. And then you extract the DNA from that soil sample or water sample, or whatever sample you're interested in. And then you can use this DNA for all kinds of interesting things. So the DNA can be used for biomarkers. So in Rachel's talk, she was looking for biomarkers of the presence of minerals. Here, when we're addressing a catastrophic disturbance, we're looking for biomarkers of the disturbance. So in this plot, here's an aerial view of the Mount Polley area, some natural lakes and the area, disturbed areas, and you can see that one particular organism, this Thiobacillus, was growing quite happily in the disturbed areas. It's a natural microorganism, it's found everywhere, but it seems to very much like the environment of of the deposited tailings. And so this is important because we can begin to understand what underlying biogeochemical processes are taking place. And knowing what types of organisms are there and getting the metagenomics piece that Stephen talked about, we can identify the good and the bad actors. And then most importantly, we can identify the microorganisms whose metabolic potential can be leveraged so as to remediate the area, the mined and the disturbed sites, and allow for ecological restoration. So this is just an example of bacteria that could be considered bad and good in the context of a mine environment. They're not always, I'm not saying they're always good or always bad in other contexts. There are many places where both of these groups are very important. So in a mine environment, sulfur oxidizing bacteria can cause sulfide minerals, which are solids, to dissolve. And in so doing, release potentially toxic elements such as arsenic and create a very hostile environment, such as acid. On the other hand, in many mine environments, sulfate-reducing bacteria are considered good bacteria. They take sulfate and reduce it into sulfide, which reacts with these metals in solution, causing them to precipitate out as sparingly soluble metal sulfides. So the objective in mine remediation is to engineer our systems in such a way as they favor the good bacteria over the bad. So it's always best to start mine remediation at the source. So for instance, coal mining waste can leach out an element called selenium that can be very damaging to aquatic life. So other than just stopping mining altogether, it is best to prevent the release of selenium from the waste rock piles. That involves learning about what types of bacteria already exist in the coal mine wastes. Are they good or are they bad? Where do the good bacteria live? And can we transfer them to replace the bad bacteria that are accelerating the release of selenium? By engineering the waste rock environment, we can take action to prevent the bad bacteria. For instance, limiting the oxygen that they might need to breathe. Or we can create an environment that favors the beneficial microorganisms, doing things such as supplementing the good bacteria, bioaugmentation with the bacteria with the metabolic potential we want, encouraging the formation of biofilms, adding special nutrients and other things that can alter the conditions inside the mine waste. All of this requires knowing what the microbial community is inside the waste and how to work with it in order to shift the community towards more favorable functional traits that prevent metal leaching. So we still only know less than 1% of all the microorganisms on Earth. In order to leverage the metabolic potential of microorganisms that can aid in mine remediation, we're prospecting for microorganisms at mine sites to select for consortia that have the capacity to perform the functions that we desire. For example, such as bacteria that can change selenium from soluble forms to insoluble forms and pull the selenium out of the mine-influenced water. So to know exactly who is capable of doing what, we use the metagenomics tools that Stephen told you about at the very beginning of this talk. That allows us to take the DNA from our sample, and then just from that DNA, we can assemble genomes for each of the species that are in that sample. And then we have a good idea of what microorganisms are there, and not only that, what are the functions that they can perform. And that might be functions important for mine remediation, but it also be functions that are important to know how to cultivate them. And I should also note that we're often looking at consortia of microorganisms. It's not one individual species that can achieve all of these goals. It's a group of microorganisms working together in a community. So despite our best efforts to prevent metal leaching, mine-influenced water can be formed on mine sites, like the acid rock drainage that Vikram mentioned. And again, we have the opportunity here to leverage the metabolic potential of the mine microbiome in order to engineer treatment systems for removing the toxic components and rendering the water safe for release. There are many different types of engineered systems that can be used, from so-called active processes that use tanks, pumps, process control, to so-called passive treatment systems that leverage natural biogeochemical processes. In many cases, the required microbial communities with the desired traits are already in the water to be treated, and within the engineered environment, conditions are created to favor those that contribute to treatment. Sometimes these systems don't work properly, and then we can use metagenomics as a diagnostic tool to understand what the metabolic potential looks like when they're working properly, and what it looks like when they're not working properly, and do that comparison that Stephen showed, and then we can tease out the actual causes for that disturbance and do something about it. So what is the future for mine remediation? So we're making use of opportunities to interact with other industries. And here we can use residues from other industries on the mine site to do mine remediation. So this diverts these residues from being landfilled, which is what is happening currently, where they pose a liability to the companies. Also, we are developing new hybrid active-passive mine-influenced water treatment systems, specifically so that we can recover values from the mine water, such as metals and also nutrients. This not only removes these liabilities from the mine site, and it also enables their reuse in other applications. So overall, mine water treatment can produce clean water for reuse and therefore reduce the dependency on the ever-diminishing freshwater supply. So these new hybrid active and passive treatment systems can be created with the elements of process control to properly leverage the metabolic potential of mine microbiome or the metabolic potential of the synthetic microorganisms that Vikram is going to create. So that's all for me. Thanks very much for listening, and I'm really delighted to answer any questions that you may have.
Thanks very much, Sue. Great presentation, and also I guess the vision for mining being part of a circular economy. It's really the holy grail for our industry. So we've got some time for questions. Perhaps Dana, if I could ask you to moderate the questions.